Welcome students to yet another class of YouTube live classes in botany. So today we shall continue with our chapter respiration, which we had started in the last class. So in the previous class we had discussed about the mechanism of aerobic respiration starting from the process of glycolysis. So recalling what we had studied in the last class, so glycolysis was the first basic step common to both aerobic and anaerobic respiration in the organisms. So glycolysis, as we had seen, means the breakdown of sugar or glucose. So previously, in the previous slide, we can recall what we had seen in glycolysis. So in glycolysis, we saw that glucose molecule, the sugar 6-carbon sugar molecule, which is the respiratory substrate or the source of energy for us. So glucose molecule was broken down in a series of 10 steps into 3-carbon 2 molecules of pyruvate. So pyruvate is the compound that is common for both aerobic as well as anaerobic respiration. So if pyruvate, what is the fate of pyruvate after it is formed from glycolysis? So there can be different fates of pyruvate. If there is, this is pyruvate, breakdown of pyruvate, the fate of pyruvate depends on the presence or absence of oxygen for respiration. In the absence of oxygen, what is formed from pyruvate is ethyl alcohol or lactic acid, which we saw in anaerobic respiration. The amount of energy released is only 2 ATP molecule, that is from the direct process of glycolysis. So if there is aerobic respiration, then what happens to pyruvate? So if there is aerobic respiration, so you can see this is pyruvate molecule and a carboxyl group is removed from pyruvate, releasing carbon dioxide. So one molecule of carbon dioxide is released in the first step where carboxylation, decarboxylation takes place. So here the second step is, it is an oxidation reaction. So here we say this type of reaction is oxidative decarboxylation. That is, carboxylation, decarboxylation takes place, resulting in the removal of one molecule of carbon dioxide, and also the oxidation process takes place where hydrogen is removed and it is accepted by the molecule or cofactor NAD. So NAD becomes NADH, carbon dioxide is released, and also there is addition of another molecule, that is coenzyme A. So NAD plus here is reduced to NADH. Ultimately what we obtain as the product is 2-carbon acetyl coenzyme A. So here coenzyme A gets attached. So this is the acetyl group with 2 carbon and this is acetyl coenzyme A. So an acetyl group is transferred to coenzyme A, resulting in the formation of acetyl coenzyme A. So acetyl coenzyme A is formed. So here you find the acetyl group is formed. So acetyl group is transferred to coenzyme A, resulting in the formation of acetyl coenzyme A. So acetyl coenzyme A is formed in this process. So what we get is acetyl coenzyme A. So what happens to acetyl coenzyme A? The next part of the respiration process is our TCA cycle or the citric acid cycle. So in TCA cycle or citric acid cycle, what is the set of reactions that will occur? So taking into account what is TCA cycle or Krebs cycle. The TCA cycle or the Krebs cycle is tricarboxylic acid cycle or the Krebs cycle. This cycle was worked out by Hans Adolf Krebs from 1932. He completed the work in 1937 and he worked on pigeon liver cells which is rich in mitochondria to find out the steps of the TCA cycle or the Krebs cycle. So, why it is known as TCA cycle is because the first product is a tricarboxylic acid or citric acid, and Krebs cycle is named after him. So, Hans Krebs also won the Nobel Prize in 1953 for finding out the process of TCA cycle or the different steps of TCA cycle. So, where does TCA cycle occur? This TCA cycle occurs in the matrix of the mitochondria. So, we saw glycolysis occurred in the cytosol or cytoplasm of the cell and does not require the mitochondria, whereas this part, the TCA cycle or the Krebs cycle, will occur in the matrix of the mitochondria. So, we can see where this TCA cycle operates. So, TCA cycle will operate in the matrix of the mitochondria. So, the matrix of the mitochondria will be seen. So, we can see the structure of mitochondria. So, if this is the mitochondria structure that you have already studied, let us see the different parts where glycolysis is occurring. So, glycolysis occurred in the cytoplasm. Glycolysis occurred in the cytoplasm. This TCA cycle will occur in the matrix of the mitochondria. So, this is the outer membrane of mitochondria, inner membrane of mitochondria, and these are the crista or the cristae membrane with F0, F1 particles attached, and this is the matrix of the mitochondria with the enzymes of the Krebs cycle or the TCA cycle. So, that is why it is also known as citric acid cycle, because the first compound that is formed is citrate. So, looking at the reactions that occur for TCA cycle, the first reaction that we had seen, that is pyruvate molecule taking up coenzyme A, losing a molecule of carbon dioxide and also getting oxidized, where hydrogen is accepted by NAD plus, forms the molecule acetyl coenzyme A. So, what is formed is acetyl coenzyme A. Acetyl coenzyme A enters into the Krebs cycle. So, the different reactions of the Krebs cycle can be studied one by one, where we see that carbon dioxide will be removed and oxidation results in the production of NADH and FADH2 molecule. So, going to each step of the reaction. So, the first reaction here of the TCA cycle is condensation of acetyl coenzyme A with oxaloacetate. So, condensation of acetyl coenzyme A with the molecule of oxaloacetate. So, acetyl coenzyme A, as we see, is a 2-carbon compound. The acetyl coenzyme A was formed from pyruvate molecule, and the next compound that we will take into account is 4-carbon oxaloacetate. So, here you can see that this molecule is acetyl coenzyme A. Acetyl coenzyme A, here you see this is CH3 C double bond O with a coenzyme attached, and here the enzyme that is used is citrate synthase. So, it helps in the formation of citrate molecule, and what is released here is coenzyme A. Coenzyme A is removed and this molecule becomes the citrate. You get 2 carbon from here, 4 carbon from oxaloacetate molecule. Oxaloacetate molecule is having 2 carboxylic groups and 1 carboxylic group is got from acetyl coenzyme A. So here you can notice these are 3 carboxylic groups that are found in the citric acid, or protonated form is citrate molecule. So this is the formation of the first compound, that is citrate. So citrate is the first compound, hence the reaction steps are known as citric acid cycle. So the second reaction for TCA cycle is isomerization reaction of citrate into isocitrate. So here there is also an intermediate compound which is called as cis-aconitase. So in this reaction you can see that citrate or the citrate molecule, the tricarboxylic acid molecule, with the help of enzyme aconitase is first getting dehydrated, that is water molecule is removed, and there is an intermediate compound that is formed which is called as cis-aconitase. So this enzyme or the compound is cis-aconitase. Cis-aconitase is unstable and then it gets further hydrated with addition of water in presence of enzyme aconitase. So cis-aconitase remains attached to the enzyme aconitase and is not released. So then it gets hydrated and forms the molecule of isocitrate. So this is isocitrate molecule. So this molecule is isocitrate. So this is the second part of the reaction. So the first reaction was formation of citrate. The second reaction is formation of the molecule of isocitrate. So citrate is formed by the addition of acetyl coenzyme A with oxaloacetate molecule. So what happens next is the third reaction. So in the third reaction we can see it is oxidative decarboxylation again. So the first reaction was oxidative decarboxylation when acetyl coenzyme A was formed. Again we have an oxidative decarboxylation reaction. So in this oxidative decarboxylation reaction, it is oxidative decarboxylation of isocitrate. So isocitrate molecule, what happens during oxidative decarboxylation is there will be removal of one molecule of carbon dioxide, which is decarboxylation process. There will be also removal of hydrogen, which is the oxidation process. So it will be oxidative decarboxylation process. So isocitrate molecule here, then losing a molecule of carbon dioxide, you can see the colored portion of carbon dioxide that is indicated in the figure. So there is removal of 2 hydrogen, a pair of hydrogen, and that hydrogen is accepted by NAD. So NAD is nicotinamide adenine dinucleotide, which gets reduced into NADH plus H plus. So the enzyme that is required for this removal of hydrogen is isocitrate dehydrogenase. So isocitrate is oxidized and it is transferred, hydrogen is transferred to NAD plus or sometimes NADP plus depending on the isocitrate dehydrogenase enzyme. So here again there is formation of an unstable intermediate compound which is oxalosuccinate. So oxalosuccinate is highly unstable and then releasing a molecule of carbon dioxide, the reaction forms the alpha ketogluteric acid. There is formation of alpha ketogluteric acid. So, alpha ketogluteric acid is formed in this reaction. So, alpha ketogluteric acid or alpha ketoglutarate. You can find alpha ketoglutarate is formed by this reaction. So, rearrangement of the enol intermediate will generate alpha ketoglutarate. So, coming to the structure of alpha ketoglutarate, how much carbon is now present? So, isocitrate is a 6 carbon compound losing a molecule of carbon dioxide. This compound that is formed becomes a 5 carbon compound. So, this is 5 carbon compound alpha ketoglutarate. This alpha ketoglutarate is also an important compound. In many cases, it is also utilized for the formation of amino acid glutamine. So, Krebs cycle will also provide alpha ketoglutarate for the synthesis of glutamine amino acid in this cell. So, alpha ketoglutarate here is a 5 carbon compound. The next process or the fourth step of the TCA cycle is again decarboxylation of alpha ketoglutarate. So, this is oxidative decarboxylation here of isocitrate, then oxidative decarboxylation of alpha ketoglutarate. So, alpha ketoglutarate molecule here, this is alpha ketoglutarate molecule. Again, if we have oxidative decarboxylation, there will be removal of one molecule of carbon dioxide. You can see this COO minus part is coming out as one molecule of carbon dioxide, and then hydrogen that is released is again accepted by NAD plus to become NADH molecule. Then the other process that takes place here is addition of again coenzyme A. So, coenzyme A is added on to this molecule forming the 4 carbon compound, that is succinyl coenzyme A. We have lost a molecule of carbon dioxide from alpha ketoglutarate. So, here there is 4 carbon again. So, here if you come to the reactions that we have seen up to this point, is that when pyruvate was converted into acetyl coenzyme A, there was loss of one molecule of carbon dioxide. Again, in this reaction of isocitrate into alpha ketoglutarate, there is loss of one molecule of carbon dioxide. And further, when alpha ketoglutarate is converted into succinyl coenzyme A, there is also loss of one molecule of carbon dioxide. So, here 3 molecules of carbon dioxide have been lost by decarboxylation. So, the pyruvate which contained 3 carbon dioxide or 3 carbon, the carbons are here all released. And the further the reactions that occur after succinyl coenzyme A will be to regenerate again oxaloacetate in the matrix, so that the process continues. So, what is the 5th step of this process is conversion of the molecule of succinyl coenzyme A into 4 carbon succinate. So, the conversion of succinyl coenzyme A into succinate. So, succinyl coenzyme A, the breakdown of succinyl coenzyme A is an exergonic process where the amount of free energy released is much. So, here that free energy can be utilized for the synthesis of a molecule of GTP. So, the breakdown of succinyl coenzyme A, so the breakdown of succinyl coenzyme A So, the breakdown of succinyl coenzyme here from this point, in presence of succinyl coenzyme A synthetase, the energy that is released is utilized in the formation of this molecule, that is GDP plus inorganic phosphate becomes a molecule of GTP. So, this molecule of GTP that is produced will be later on converted into the molecule of ATP. So, GTP later on gets converted into the molecule of ATP. So, GDP plus inorganic phosphate, the energy available from this molecule or breakdown of coenzyme A and the formation of GTP molecule is a type of reaction called substrate level phosphorylation. So, here the energy is obtained by the breakdown of a compound and their GTP molecule can be synthesized. So, this GTP molecule is the result of substrate level phosphorylation and this GTP is later on converted into one molecule of ATP. So, what we get as the product is here 4-carbon acid which is succinate. So, this molecule is succinate. The next step of the reaction is dehydration of succinate into fumarate. So, here succinate is again a 4-carbon compound, fumarate is also a 4-carbon compound. Here we have dehydrogenation reaction. So, here there will be removal of hydrogen from succinate molecule. So, here you can see these two hydrogen are being removed. The two hydrogen are accepted by the FAD molecule which is flavin adenine dinucleotide. So, flavin adenine dinucleotide becomes reduced by accepting a pair of hydrogen. The enzyme that is required here is succinate dehydrogenase. So, succinate dehydrogenase is the enzyme. This enzyme is also present later on in the electron transport system that we can see. So, this is fumarate molecule that is formed when hydrogen is removed from the molecule of succinate. So, these are now the 6 reactions that have occurred. The 7th reaction or the 7th step is hydration of fumarate into malate. So, fumarate molecule is hydrated, that is addition of OH will take place and this is the molecule of malate that is formed in presence of enzyme fumarase. So, hydration of fumarate takes place into the molecule of malate. The last step is dehydrogenation of the malate molecule into the regenerated compound, that is oxaloacetate. So, here this is malate and in presence of enzyme malate dehydrogenase, a pair of hydrogen is accepted by NAD to become NADH plus H plus and you are getting back the 4-carbon oxaloacetate. So, oxaloacetate is again regenerated to continue the process of TCA cycle. So, these are the 8 reactions that occur in the TCA cycle. So, coming back to the entire cycle of TCA, so here what we find is summarizing the reactions, acetyl coenzyme A combines with oxaloacetate molecule. Oxaloacetate is a 4-carbon compound, acetyl coenzyme A is a 2-carbon compound, and then it becomes, in presence of enzyme citrate synthetase, it forms a molecule of tricarboxylic citrate. So, 3 carboxylic groups remain attached. So, this was the first reaction, and here coenzyme A is released. The second reaction was citrate was converted into isocitrate, the isomerization reaction, where an intermediate product cis-aconitic acid was also produced. So, the enzyme was aconitase, we get isocitrate. The third reaction was deoxidative decarboxylation of the isocitrate molecule. So, isocitrate molecule undergoing oxidative decarboxylation releases a molecule of carbon dioxide, a pair of hydrogen is released. This pair of hydrogen is accepted by NAD plus to become NADH and H plus. The oxidative decarboxylation process here then results in the formation of 5-carbon compound alpha ketoglutarate. So, alpha ketoglutarate is formed. So, this molecule alpha ketoglutarate is 5-carbon. Again there is oxidative decarboxylation of alpha ketoglutarate releasing a molecule of carbon dioxide, a pair of hydrogen accepted by NAD to become NADH plus H plus. The enzyme is alpha ketoglutarate dehydrogenase. So, dehydrogenase enzyme, the function of this enzyme is to remove hydrogen. So, here again coenzyme A is added. So, after the addition of coenzyme A, the molecule becomes succinyl coenzyme A 4-carbon compound. So, the 5-carbon alpha ketoglutarate gets converted into succinyl coenzyme A. So, succinyl coenzyme A, the next reaction is breaking of coenzyme A from succinyl coenzyme A, and this is an exergonic process which releases sufficient free energy, and this free energy is utilized in the synthesis of GTP molecule. The enzyme here is succinyl coenzyme synthetase and a molecule of GTP is produced. This GTP will be later on converted to a molecule of ATP and coenzyme A is released. This results in the formation of 4-carbon succinate molecule. So, succinate in presence of dehydrogenase, succinate dehydrogenase, the pair of hydrogen that is released in this process is accepted by flavin adenine dinucleotide or FAD to become a molecule of FADH2, and then what is formed is 4-carbon fumarate molecule. This fumarate molecule adds up water in presence of enzyme fumarase. So, there is hydration of fumarate and it becomes the molecule of malate. 4-carbon malate is formed. The removal of hydrogen, again the removal of hydrogen from malate in presence of enzyme malate dehydrogenase forms or regenerates back our oxaloacetate molecule in the matrix of the mitochondria in presence of enzyme malate dehydrogenase. So, the entire process of TCA cycle mainly is an oxidation process where the pairs of hydrogen are being removed and they are accepted by the molecules like NAD or FADH2. So, calculating how many number of NADH molecules are obtained from the TCA cycle and what is the probability of obtaining how many ATP molecules, from these reactions. So, the number of NADH molecules, if we calculate NADH, the number of NADH molecules is, the first molecule produced is from pyruvate, to acetyl coenzyme A. So, here we get one number of NADH molecule. The second NADH molecule is from isocitrate to alpha ketoglutaric acid or alpha ketoglutarate. Alpha ketoglutarate. So, here again we get one NADH molecule. The third is from alpha ketoglutarate to succinyl coenzyme A. So, here again there is release of one molecule of NADH, and the fourth one is where malate to oxaloacetate. You get one molecule of NADH. So, the number of NADH molecules obtained here is 4 NADH molecules. What is the number of FADH2 that is obtained? So, FADH2 is from succinate to fumarate. We are getting one molecule of FADH2, and the synthesis of direct molecule of ATP is one molecule of GTP converted into one molecule of ATP in the reaction where succinyl coenzyme A breaks the coenzyme A to get converted into succinate. The probability of obtaining the number of ATP molecules when the NADH and FADH2 molecules enter into the next step of electron transport system. So, one NADH molecules can approximately release 3 molecules of ATP, whereas one molecule of FADH2 can release 2 molecules of ATP. So, the number of ATP molecules that can be obtained is 4 into 3 ATP for NADH, that is 12 ATP. For one FADH2 molecule, you can have 1 into 2 ATP molecules, that is 2 ATP molecule, and there will be synthesis of 1 ATP direct molecule. This is amounting to 1 ATP. So, here the total becomes 15 ATP molecule for one pyruvate molecule that is oxidized by the TCA cycle. So, in the glycolysis process, we obtained the 2 molecules of pyruvate, and so from 2 molecules of pyruvate, the probability is we can get 30 molecules of ATP when there is electron transport system and oxidative phosphorylation after the TCA cycle. So, this is what happens in TCA cycle, a set of 8 reactions that occur. So, after the TCA cycle, we shall come to the electron transport system of the mitochondrial chamber and what is present in the membrane system and how ATP is synthesized by oxidative phosphorylation. So, next portion of the respiration is electron transport system, and first let us see what are the components of the electron transport system, where they are found and what are the different complexes. So, the components of electron transport system are, there are a number of electron carriers which are arranged according to their increasing redox potential in the inner membrane of the mitochondria. So, these molecules will be arranged in the inner mitochondrial membrane. So, in the inner mitochondrial membrane, suppose we take, this is the mitochondria, this is the outer membrane, then you have the inner membrane with the crista. So, these inner membrane will be having the F0, F1 particles, and in between we come across all these electron carriers that are arranged according to their increasing redox potential, the ability to get reduced, and it includes, what it includes is different types of electron donors. These electron donors are molecules of NADH and FADH2 that were produced in the matrix. So, here the TCA cycle continued, we got NADH and FADH2 molecules in the matrix of the mitochondria. So, these molecules here will act as the electron donors. So, these two molecules NADH and FADH2 do not have much affinity for the electron. So, when they come in contact with the electron acceptors which have high affinity for electron, they will be donating the electron that is present. So, NADH and FADH2 will be getting oxidized when they come in contact with the electron transport system. So, it consists of the electron donors, that is NADH and FADH2. Then there is electron transport carriers and also the ultimate electron acceptor, which is oxygen present in the mitochondrial membrane. So, until now we have not seen the direct usage of oxygen, so which will be acting as the terminal electron acceptor. So, the terminal electron acceptor is our oxygen molecule that is present in the matrix of the mitochondria. So, what is present here is 4 complexes and 2 mobile carriers in the system. The electron transport system consists of 4 complexes and 2 mobile carriers. What are these 4 complexes? The first complex is called as NADH dehydrogenase. NADH dehydrogenase, that is the enzyme that is going to dehydrogenate NADH, remove a pair of hydrogen and transfer the electron to the next component. So, this is NADH. The redox groups here, what is going to get reduced or oxidized is, it has a molecule of flavin mononucleotide. So, this is flavin mononucleotide, and also it has iron sulfur components or iron sulfur centers. There are 6 iron sulfur centers in the NADH dehydrogenase complex. So, the second complex is succinate dehydrogenase. So, succinate dehydrogenase, we saw succinic acid or succinate conversion. So, there the redox molecule or redox group is FAD, which is flavin adenine dinucleotide. So, FAD and FMN are the redox groups present in these two complexes along with several iron sulfur centers. So, the third complex is ubiquinone cytochrome C reductase. It also helps to pump the H plus, and here again there is iron sulfur center present. So, cytochrome BC1 complex or cytochrome C reductase is the next complex. The fourth complex or the last complex present is cytochrome oxidase, cytochrome AA3, cytochrome oxidase. It also has copper along with iron in the complex. So, here copper can also be reduced and oxidized along with the iron component in cytochrome oxidase. The two mobile carriers that can move in the membrane in the electron transport system. So, they are, one is cytochrome C, the other is ubiquinone or coenzyme Q, which is the non-protein component present. So, these two are mobile carriers which are helping to moving the electron from one complex to another complex where the complexes remain embedded or on the membrane. So, these are the four complexes. So, the last part here will also include your F0, F1 particle or the ATP synthase, which takes part in the synthesis of ATP molecule. So, we can have a look at the different arrangement of how these complexes are arranged in the membrane. So, have a look at the electron transfer system. So, you can see this is the upper part, shows you the outer membrane of the mitochondria. So, this is the upper part of the mitochondria or the outer membrane. Then you have the intermembrane space, there is intermembrane space that is present. So, this is the outer membrane, then this is the intermembrane space of the mitochondria. So, recalling the structure of mitochondrion, you can also see it in the figure. So, this is the outer membrane that is represented by this outer structure. Then this is the intermembrane space. This space is the intermembrane space and this represents the inner membrane. So, this is the inner membrane part, this is the outer membrane part and this is the intermembrane space that is present in the mitochondria. So, here you can see that the electron transport complexes are present in the inner membrane. So, this is the first complex that we talked about, NADH oxidoreductase or NADH dehydrogenase, that is going to remove hydrogen from NADH molecule. So, this is the first complex. The second complex remaining embedded in the inner membrane towards the matrix side. So, this reaction was occurring when succinic acid got converted into fumaric acid or succinate to fumarate. So, this enzyme remains embedded towards the mitochondrial matrix side and this is the second complex that is present. The third complex is the cytochrome BC1 complex or cytochrome C oxidoreductase. So, this cytochrome C complex organization of the complexes in the inner mitochondrial membrane. So, So, inner mitochondrial membrane has the arrangement of these complexes, and then ultimately here you can see the ATP synthase, also sometimes considered as the fifth complex, the F0, F1 particle that will help in the synthesis of ultimately ATP molecule. This is the matrix side where we find oxygen is present in the mitochondrial matrix. So, the synthesis of ATP molecule, which is called as oxidative phosphorylation. This process of synthesis of ATP shall be discussed in the next class of respiration. So, in the next class we shall see how ATP is synthesized in this electron transport system, how it becomes the terminal acceptor of electrons, and then the what is respiratory quotient and the different factors that affect the process of respiration. So, for today this is the end of the class, and so you can go through the electron transport system and the Krebs cycle and the part of glycolysis for respiration.